Hi everyone, welcome to our video. Today we're diving into one of the most exciting frontiers in science, synthetic biology. Imagine being able to program living cells the way we program computers. Sounds wild, right? But that's exactly what scientists are doing, engineering life with code. Over the next 15 minutes, I'll be chatting with my friends about how synthetic biology works, why computation is changing the game, and what it means for the future of medicine, energy, and even ethics. We'll keep it casual with some fun props and skits along the way, so you'll see how biology isn't just about pipettes and petri dishes anymore. It's about algorithm, models, and creativity. So let's get started. First up, I'll be talking with Aina and Anur about the trend of synthetic biology and the computational tools that make it possible. Hi everyone, I'm Hanis and today I'll be interviewing Aina and Anur about synthetic biology and computational tools. This field basically turns science into experiment and play to let the computer suffer instead of us. So let's get started before my brain crashes like my laptop. So here we have Anur and Aina. So Anur, what is synthetic biology?
Synthetic biology is a field where we design and build a biological system for specific purpose. Okay, instead of only studying natural biology, scientists engineer gene, protein, and pathway to perform a useful function like producing medicine, biofuel, and detecting disease.
So what is the difference between traditional and modern synthetic biology? And why is modern is more preferred than the traditional one? Honestly, biology used to be very trial and error. Scientists will often try one condition and fail, and if they try again, and they often to fail again. Back in the day, people even call this as a pipette and pray method because sometimes you pipette one thing and you pray for it to work. The condition is not certain.
So Anur, why is that approach not effective?
Because it's time consuming, costly, and unpredictable. So you may repeat the experiment many times but it still fails. So that's why the scientists started using the computational tool to guide the experiment.
So why do we need computation in synthetic biology?
Because biology is very complicated. If you can see only one cell, there's actually 1,000 genes interacting on it. Okay, come here, take a look. Okay, for example, by using this prot, you can see a lot of genes interacting in one protein. So, by using computer, we can avoid and prevent from wasting money, wasting time, and wasting energy from finding only one thing we needed. So this actually is not lazy. This is smart.
Oh my god! Wow!
So what role do computers play when planning or predicting experiments?
Computer allow scientists to design and test. So they help predict the whether a gene edit will work or whether a protein will fold correctly and whether a pathway is efficient. So lab work become more targeted and accurate.
Aina, what's a simple example of protein design algorithm?
Okay, for example, imagine you want a protein that can break down a plastic. But you need a lot of experiment to find the protein. So instead of randomly testing thousands of proteins in the lab, we use the computer to design the protein first. We let the computer suffer. Software predicts the shape, folding, and function of the protein. So when we finally go to the lab, we already know this is the one that we needed. High chance of working, less praying, and more predicting.
Anur, what are genome design system?
Genome design system help scientists plan and edit DNA sequence digitally. For example, when designing bacteria to produce insulin, the computer checks if all genes work well together before actual genetic modification.
What is metabolic modeling?
My little soda pop.
Do you know actually metabolic biofuel is like a Google Maps for a metabolism? If we want a bacteria to produce biofuel, the computer stimulates different pathway, and of course the computer will give you the best one and tell us which route gives the highest yield. So we don't take the long road, but we take the most efficient one.
So Anur, how do computational tools help CRISPR avoid cutting at the wrong spot?
CRISPR can sometimes cut DNA at the wrong place. and call off a target effect. So computational tools predict this off-target site and making gene editing safer and more precise.
What are gene circuits and how does the computation help design them? So, gene circuits are basically electric circuits but biological. Using computational tools, we can design genes to switch on or off under a certain conditions. For example, insulin is only produced when glucose levels are high. So cell behaves like a smart device, not random.
Wow, thanks Aina and Anur. That was awesome. So we've seen how computation helped design protein genomes and even gene circuits. But here's the big question. Once you design something on the computer, how do you actually build it in real life? Well, Lai and Irene are working on exactly that. Let's check in with them and see how the design build test learn cycle works and why ethics matter just as much as algorithms.
Hey guys, what are you guys working on? It looks intense.
Hi Anis, we're currently setting up an experiment where we engineer bacteria to produce insulin.
Insulin, like for diabetes, you can make that in bacteria.
Absolutely. It is one of the oldest and most successful example of synthetic biology. The real magic today happens before we even step into this lab, on the computer.
Really? How does that work?
Well, synthetic biology has moved far beyond trial and error. Now, bioinformatics and computational design are essential. We use digital tools to model, predict, and design biological systems before we build them. For our insulin project, we didn't just insert random DNA. We used protein design algorithms to optimize the insulin gene sequence for bacterial expression and metabolic modeling software to predict how adding this new function would affect the bacterium's health and production yield. It's all about precision design.
Oh, I see. So you design it digitally first, but how do you go from a computer model to the real thing, given how unpredictable biological processes can be?
That is the key challenge. This is where design, build, test, learn cycle come in. It is the core feedback loop of synthetic biology.
Design. That is what Lai just described, using software to plan the genetic construct. Second, build. We physically assemble the DNA and insert it into the bacteria in the lab. Third, test. We run experiments to see if the bacteria actually produce insulin and how much. Fourth, learn. We analyze the test data. Did it match our computational model? If not, why? The problem with the traditional cycle is that the build and test phases are slow and expensive. You might wait days for cells to grow, only to find the design fail.
This is where computation dramatically speeds things up. Advanced circuit simulation tools can run thousands of virtual test cycles in minutes, predicting how our genetic circuit will behave. Machine learning models can analyze all our past learned data to suggest better design for the next round. It compresses years of lab work into months or weeks, allowing for rapid optimization.
Wow, computation has made synthetic biology faster than ever, but it's also raised ethical concerns. Like, are these concerns justified?
If the models work and the data looks good, I don't see the problem. Science advanced by moving fast.
Wait a minute!
That confidence is exactly the concern. Computational models are not reality. They are the simplified guesses of extremely complex biology.
But they are statistically accurate.
So are weather forecasts, and we still get surprised by the storm. Cells don't always follow prediction, especially outside control conditions.
What could actually go wrong?
You can get off-target effect. The gene designed for one function may unintentionally affect other pathway.
Unexpected results are part of science. That's how discoveries happen.
In living system, unexpected can mean toxicity, system failure, or damage beyond the lab.
Are there broader risks beyond the lab?
Yes, biosecurity. The same computational tools that design beneficial organisms can be misused to design harmful ones.
But it's just a software.
Software that design living system. That is no longer virtual. It is physical and irreversible.
Five minutes later.
So what's the solution then?
Human oversight everything. Ethical review, safety testing, and regulation must guide every stage of the DBTL cycle.
Fine, maybe the algorithm shouldn't be making life and death decision.
Exactly. Technology should inform choices, not replace responsibility. So, we both agree that in synthetic biology, progress without ethics is not innovation, it is a risk.
Wow, thank you guys. It's fascinating to see how computing is revolutionizing biology, but it's clear that we have to be incredibly thoughtful and responsible with this power. I learned a lot. Thank you.
Welcome! It is a thrilling field to be in.
And it's only going to grow with future design AI organism and fully automated labs pushing through the boundaries of what's possible.
Wow, wait, what a journey! We've seen how synthetic biology is transforming from trial and error experiments into a design driven science powered by computation. From protein design algorithm to the DBTL cycle, it's clear that computers are now as essential in the lab as microscopes. But we also learned that with great power comes great responsibility. These tools can speed up discoveries and create life saving innovations, but they also raise important ethical and safety questions. So, the takeaway synthetic biology is not just about engineering life, it's about engineering responsibly. So thanks to Aina, Anurulai, and Irene for sharing their insights. And thank you all for joining us. Synthetic biology is still young, but it's growing fast, and who knows, maybe one day you'll be the one designing the next breakthrough organism. Until then, stay curious, stay creative, and keep exploring. Bye!